Hello and welcome back. My name is Carl Geiser. I am a research associate here at ISB, and it is my absolute delight to be here. I'm looking forward to chairing this session, and we're going over the commensal host interaction in diseases today. And to that effect, we have two fantastic speakers on this topic, Dr. Arjun Raman and Dr. Anna Weil. Just a reminder that these talks will be back-to-back. We will have a panel discussion after both speakers have had an opportunity to present. So if you have any great Q&A questions, please save them, put them in the Q&A button, and please remember to address the speaker when you ask the question, so we know who to point the question out. So if I could ask Dr. Raman to share his screen. Dr. Raman is the assistant, I'm sorry? Okay, one moment, just a quick technical difficulty. Okay, that should be better. All right. Yes, Dr. Raman is currently an assistant professor of pathology at University of Chicago, which is where he got his start. He earned his Bachelor of Arts in Physics and his Bachelor of Science in Chemistry there before moving on to the University of Texas Southwestern, where he worked in the Raghunathan lab, applying statistical genetics to help identify residues of protein function. After he earned his doctorate in 2017, he then moved on as a postdoctoral fellow in 2020 in Gordon's lab at Washington University in St. Louis. And there he used applied analytical techniques to create mathematical representations of microbes important to gut microbiome function and adaptation. His current lab explores these underlying principles of structure function of these complex biological systems. And I think he has a wonderful talk for us today going over the statistical design of the functional microbiome. So go ahead and take it, Dr. Raman.
Thank you very much. Can you guys see my screen? I just want to make sure that this desktop is the right one. Like you can see a title.
Yes, but it's not in presenting mode.
It's not in presenter mode. How about now? Now it's in presenter mode. Okay, great. Thank you very much for that kind introduction. Thank you to the organizers for inviting me. I'm really excited to share what is an unpublished story from our lab and really work that was done by one fantastic postdoc, Rita Oliveira. So before I get into it, perhaps I could, my lab started in in fall of 2020. So we've been at the University of Chicago for on the order of 3 years within the pathology department, but also in a microbiome center called the Duchossois Family Institute that is geared towards understanding the human gut microbiome for augmenting human health. And what I'd like to do is tell you a little bit about what my lab does and then how we got into the journey of trying to create synthetic microbiomes for health purposes. So to start, we have been probably since 2010s we've been thinking about synthetic biology as a fourth technological revolution and the immense promise that it holds for solving lots of problems, not just in human health, but in climate tech and agricultural health. But turns out that it's like very hard to perform synthetic biology in any scalable way. And when we boil this difficulty down to maybe perhaps a fundamental reason, it's because these kinds of systems that nature has created are incredibly complex. So here is an example of such a system, E. coli, that we're all familiar with. It contains thousands of genes, and amongst those contains millions of gene interactions. because the function of whatever phenotype we care about generally comes from not just a single gene, but the emergent architecture of many genes that interact with each other. So as a result, because we currently don't have frameworks for emergence, there's no statistical framework to describe how parts interact together to create collective wholes without invoking some sort of grand designer. Our current, quote, design principle for synthetic biology can really be boiled down to trial and error. We swap out parts or we mutate parts and we see what works, and we perform high throughput phenotypic screens to finally get at a solution if we are able to arrive at one. So one of the central thrusts of our lab is to try to understand rules of emergence. That is, how do individual parts, whether those parts are amino acids that create a protein or genes that create a genome, or in the case that I'm going to talk about today, bacteria that create microbiomes, can we learn rules of biosystems engineering that can make this process far more scalable and perhaps more precise with respect to specific target functions that we would like to design towards? So, like I said, today, I'm going to be talking about one project in the lab, which has been our attempts at trying to create design principles for designing synthetic microbiomes against specific target functions. And what I'd like to start with is what the current design principles are. So here I have a list of bacteria, and I think with all of the technological advances that have happened over maybe the past decade or so, we are now able to high throughput phenotype bacteria to a remarkable degree. We can genome sequence them, we can metabolomically profile them, we can figure out what they grow together with. And as a result of advances in culturomics and in multi-omic profiling, we can do things like start putting together bacteria, combining them based on specific features. And what I'm showing you here is a schematic where we combine, let's say, Longum, Prosnitzii, and Ahedrus together, and we hope that collectively they do something based on their individual features. But inevitably, I think more often than not, what we find is that it's very difficult to predict what the consortia is going to do relative to the individual bacteria. I don't think this is necessarily a surprise, but the question is, how does one contend with complexity and emergence in these kinds of systems? Because again, going back to what I said earlier, because we have no framework for predicting what's going to happen based on the individual parts, this becomes a fundamentally intractable problem. The number of combinations that one needs to probe in order to arrive at what are the correct combinations of bugs, given many more bugs than one could choose from, becomes quickly astronomical without constraint and models guiding how to build such consortia. So our approach was to, I say here, use the same engineering process as nature. And what I mean by that is that evolution has also faced the problem of complexity, but has managed to converge on solutions in finite time without like burning the ocean, so to speak, without needing infinite resources to do so. And how? The question is how? Well, there's one sort of grand design principle of nature, which is selection and variation iteratively. That is, you create a system, you vary it in a specific way, select, see what happens, and then iterate back. So we actually tried to do this in the lab with the idea of creating consortia. So the basic premise was we would take a strain bank, which would be our parts list, so lots of different bugs. We would design lots of consortia, we would build them, we would test them in the lab for a specific function. We would annotate each community by whether or not the function was achieved. And then we would apply statistical learning principles, machine learning and artificial intelligence, to try to now understand the constraints that would specify a functional consortia. Then we would go back and design, build, test, learn, design, build, test, learn. And in this sort of instantiation of evolution, the only wrinkle that's added here is that we would learn a model explicitly rather than continue to go through this cycle without maybe reflecting on what we had done through the design, build, test framework. I would argue that what evolution does is learn through fitness. That is, whatever exists is the thing that is now going to be able to propagate to the next generation. So what does this critically do that allows potentially a huge advantage relative to what I'll call bottom-up design? Well, it strips away the need to know anything about how any of the parts work individually, how they interact, or how function is manifest. So an alternative to this would be that if we instantiated a function like I want to kill a multi-drug resistant pathogen like Klebsiella pneumonia, I would need to know extremely intricate details about how to do that. In this formalism, one does not need to know these things. The contra, the downside is that a priori, you don't know how many cycles you need to go through. One may need to go through like 100 cycles, in which case this whole strategy would also be defunct. So we decided to undertake this strategy, and the test function that we decided to go after was killing multi-drug resistant Klebsiella pneumonia. Klebsiella pneumonia is a pathogen that overgrows in the setting of prolonged antibiotic exposure in the clinic. It's a World Health Organization priority 1 pathogen, which means that the antibiotic resistance of these bugs is increasing at a rate that's critical. And the specific strain that we decided to use for instantiating this formalism is a strain called MH258, which is a strain that is known to be multi-drug resistant. It has a carbapenemase gene in it, and it was isolated from a patient at Memorial Sloan Kettering who showed signs and symptoms of multi-drug resistant Klebsiella pneumonia infection. So where do we start with trying to do this? Well, the first thing that we need is a parts vault, so to speak. As part of the Duchossois Family Institute at the University of Chicago, we have access to a large bank of commensal bacterial strains that were isolated from 28 healthy human donors. And by here, what we mean by healthy, it's always difficult to define, but we screen for prior antibiotic use, obesity, and a slew of other clinical indications to ensure that these people had commensal strains and not pathogenic strains. We isolated these strains and whole genome sequenced them, and in total, the strain bank that we used for our design, build, test, learn strategy comprised almost 700 strains. What you're seeing on the right here is a phylogenetic classification of those strains based on 16S sequencing. And you can see that we're heavily, like a normal gut microbiota, we're heavily, or the normal statistical distribution of human gut microbiotas, we're heavily biased towards the lactose and Bacteroideaceae, with a slew of other families of bacteria that are commonly found in human guts. So imagine one now opens a freezer, sees 700 tubes, each one carrying one of these bacteria. The blind design space, and what we mean by that is if each bacteria is considered its own independent source of information, the total design space is 2 to the 700. So this is clearly not a tenable metric. It's a gigantic space verging on infinite. Doesn't matter how many liquid handling robots one has, we will never be able to cover this space. And so the first thing that we wanted to do was employ a model of constraint. So we created an alignment, a genomic alignment of all of these bacteria based on their orthologous gene groups, and then performed a UMAP-based decomposition of that alignment. So all a UMAP is called a uniform manifold approximation projection. All it is clusters. So anywhere you see a circle here, that's one of the bacteria in this pie chart. And the distance here doesn't mathematically mean anything like it would in PCA, but what it does provide is some sort of definition of relatedness between two strains. And so the first thing that we found was that though we had 700 strains in our strain bank, in fact, it was a very low dimensional projection onto a space that told us that really we have a handful of clusters that that represent metagenomic diversity. So the first thing we did was to say, well, what we don't want to do is design a whole bunch of consortia that are the same consortia, the same bacteria over and over again. Another way of saying that is our consortia shouldn't include just E. coli, E. coli, E. coli, E. coli. What we wanted to do was maximize functional non-redundancy. And so the way we did that was by utilizing the UMAP to design a set of 100 consortia based on sampling the UMAP space in an equipartitioned manner. So imagine we pick a seed bacterium randomly. The constraint for its partner would be that it needs to be maximally away from the seed bacterium in the UMAP space. And then bacteria number 3 needs to be maximally away from the first 2 clusters in the UMAP space, and so on and so forth. And this is how we would make a consortia that, in this case, what you're seeing on the screen is a 5-member consortia. And if you see the words DMC, the letters DMC, that means Design Microbial Consortium. So to tell you what we did, we constructed 96 consortia. We picked 96 because there's a 96-well plate, so it was purely practical constraint. We built 96 consortia, and we built them across a possibility of 46 strains. And the reason we picked 46 strains is because those 46 strains spanned the diversity of the UMAP space that we see here with the totality of the 700 strains in our strain bank. And you can see here the compositional matrix of each of those consortia. So anywhere, again, you see DMC, it's a Design Microbial Consortia with a number attached to it. Every one of the consortia was a subset of the 46 strains that we could have chosen. So at the very top, you see DMC 46. That means that it had all 46 strains. That consortia had all 46 strains in it. And we created a distribution of 96 where the average number of strains in a given consortia was 14, spanning from 3 to 46. Then what we did was we co-cultured each of these consortia in BHIS media, brain heart infused media with cysteine, with Klebsiella MH258 Klebsiella pneumonia strain. And we sampled every 24 hours. And what you see on the right is the time course of sampling. So we co-cultured these communities with Klebsiella pneumonia for 120 hours. And then we plated to see whether or not we could see KP growing on plates. All of this was done anaerobically because all of these commensals are all from the gut. And so a majority of them are obligate anaerobes. Okay, so what I'm showing you here is a test of all of the, is the result of testing all of the consortia against suppressing Klebsiella pneumonia in vitro. And that's shown on the green bar graph on the right. And what you can see is that there's really like a continuum of Klebsiella pneumonia suppression across all of these consortia. And what you also see is that it doesn't really correlate with complexity, which is on the complexity of the consortia, which is on the gray bar. So another way of saying that is that it doesn't matter if the consortia has a lot of strains or a few number of strains. The number of strains that the consortia has doesn't map to how well it performs. So next, what we did was we split this data set into a training and validation data set. And so we trained a very sort of standard machine learning model. This is a random forest model on 70% of this data, and then trained on the and validated on the remaining 30%. And we got good statistics for predictive capacity. But then what we did was we tested our model on the ability to predict the efficacy of new consortia, meaning consortia that are not within this 96. We created 60 new consortia. They abided by the same probability distribution as before, a span of sizes ranging from 2 to on the order of 30 members. And what I'm showing you here is on the x-axis what our RF model predicted the KP, the end KP load would be, and on the y-axis what we actually found when we co-cultured these communities for 120 hours together. What we found was a remarkable correlation between the y-axis and the x-axis, to the point where we could almost directly predict how well a community would suppress Klebsiella pneumonia without knowing anything about the dynamics in between. The only thing that went into this model was the input structure, the ones and zeros that are in this matrix. So the result number one was that in just one round of design, build, test, learn, using the design strategy that we did, which was maximizing UMAP diversity, we converged on a model that could predict the suppressive capacity of a complex consortia on Klebsiella pneumonia. So one of the questions that we then had was, what are the constraints on consortia function? The RF model doesn't tell us what are the number of numbers we need to synthesize maybe a sparse design microbial consortia. And why this is an important question is because typically we think about strain dropouts as the way to do this. But strain dropouts don't tell us about emergent capacities or higher order cooperativity amongst complex consortia. So we actually used our model to train to score 100,000 consortia in silico. And as a result, what we got was a histogram. That's what you see on this plot. And what we did was we took a threshold and we said anything that lives, any consortia that lives on this side of the histogram is a consortia that works particularly well. So, let's see. We then created a matrix of all of the consortia that were predicted to work such that they suppressed Klebsiella pneumonia to a significant degree. We again made a presence-absence matrix of this. And then what we did was defined patterns of covariance that mapped to Klebsiella pneumonia suppression. So the way we did that was we performed a technique called singular value decomposition on the matrix that you see on the left. And we asked which eigenvectors map to predicted Klebsiella pneumonia suppression the most. And what you're seeing here is on the x-axis, principal component 46, which I don't think anyone ever looks at, but turns out that was the one that most correlated with predicted KP load. Principal component 46 held less than 0.01% variance, but mapped very nicely onto how much Klebsiella pneumonia was predicted from our model to be at the end of 120 hours of co-culture. And what I'm showing you here are the projections of each strain onto principal component 46. And one could think that maybe we could use this information to try to design a consortium, but the problem is when you look at this, you see just kind of like a continuous distribution of all 46 strains. There's no real hard line with respect to what interactions between these bugs are important for us to then go and try to build. So instead, what we did was we said, well, what is the nature of how two bacteria co-project onto principal component 46, meaning what is their similarity of projection onto this axis that seems to be a particularly good axis to now identify function through. And what we found were these distinct blocks of consortia. So what you see in the top left are a group of 5 bugs, and what you see in the bottom right are a group of 10 bugs. And you see that they co-vary with each other across, right, on the off diagonal. And then you see this large block in the middle. Turns out that the top left and the bottom right are the top 15 bacteria in the projections of principal component 46. And so we thought this would be a particularly useful block to make. So I should also say that this provides a sense of which other blocks to make too. So we made all of the blocks that you see in that middle red major block. We made 3 of those blocks as well. So result number 2 is that the inference provides insight into what consortia to build. We tested each one of these blocks in a variety of different environments. So first in BHIS, what you see in gray is the 46-member consortia, meaning that it has all 46 members. And block 1 is the top left. Block 5 is the bottom right. And then what you see in dark blue is block 1 plus 5. Blocks 2, 3, and 4 comprise the large block in the middle where projections onto principal component 46 are not significant. So in BHIS, what we find is that block 1 plus 5 works quite well. We then assayed these blocks in 2 other in vitro environments that are completely different, which are germ-free mice and antibiotic-treated SPF mice, where cecal contents of these mice were scraped after sacrifice, filtered for their bacterial contents, and then made into media. So these conditions are very different than BHIS. And what we find is that across these different media conditions, the only consortia that worked well to suppress Klebsiella pneumonia was block 1 plus 5, this 15-member consortium. And so we call this SYNCOM 15, a 15-member synthetic consortium. We then tested this consortium in a murine model of infection. We had C5J black 6 mice that were exposed to broad-spectrum antibiotics, metronidazole, neomycin, and vancomycin for 4 days. Then these mice were then infected with Klebsiella pneumonia MH258, were given 3 days, and then were given either saline, which is shown in gray, a fecal microbial transplant, which is a whole stool transplant, shown in black, SYNCOM 15, shown in green, or we call it DMC 19. It's one of those blocks that is predicted to not suppress Klebsiella pneumonia. We then let these mice grow for 21 days prior to sac, and anywhere you see a brown dot here, it means we took a fecal sample and we assayed for Klebsiella pneumonia abundance in the fecal microbiota. So what I'm showing you here is on the x-axis, days post Klebsiella pneumonia infection in these mice. On the y-axis is the amount of Klebsiella pneumonia that we see. What you see at the very beginning is that all of the mice across the cohorts have a large amount of Klebsiella pneumonia. They are then triply gavaged with one of the 4 indications that you see in the color key, where DMC 15 is our SYNCOM 15. And what we found was quite remarkable. First off, the PBS treated cohort showed almost a constant Klebsiella pneumonia after day 6 at approximately 10 to the 8th load. Whereas the FMT showed us a rapid drop. You see FMT in the gray, showed a rapid drop in Klebsiella pneumonia abundance. What we found was that DMC 19, the consortia predicted not to work, indeed didn't work, looks like saline. But the green consortia, SYNCOM 15, worked quite well. In fact, worked more rapidly than a fecal microbial transplant, was less variable if you look at day 9 through day 17, and was stable in terms of suppressing Klebsiella pneumonia. So what we concluded from this was that SYNCOM 15 suppressed Klebsiella pneumonia across these diverse contexts, including in a murine model of infection. So then the final thing I'll touch on is, how does SYNCOM 15 work? We did metabolic profiling of SYNCOM 15, both in vivo and in vitro. And the bottom line is that the way it works is through short chain fatty acid production, as well as amino acid depletion. So what I'm showing you here are all the features that we found that differentiated consortia that worked particularly well from those that didn't. And what you see is that the features that we pulled out were highly enriched for short chain fatty acid content, and then phenylalanine and alanine depletion. And what I'm showing you here are the features that SYNCOM 15 specifically showed with respect to being enriched relative to the PBS cohort in the mice. If you look at phenylalanine and alanine, what we found was that SYNCOM 15 substantially suppressed phenylalanine and alanine concentrations, and also increased hexanoate concentrations relative to the consortia that did not suppress Klebsiella pneumonia. some of the other short chain fatty acids, one could argue, didn't really do much to. So one of the mechanisms that we're continuing to try to suss out is we think that the way that this consortia works is by depleting amino acids and acidifying, potentially acidifying the environment, but certainly by producing short chain fatty acids that could potentially also involve signaling to Klebsiella pneumoniae to suppress it. So what I'd like to conclude with is that I think what we've done is shown that this kind of rubric could work for designing microbial consortia without knowledge about the parts, just beyond just the genome sequence of the strain bank that we have. And what we're testing right now are a few questions regarding generalizability. You'll notice here there was nothing specific about Klebsiella pneumoniae. We could swap out the test function for any function we like, really. And what we also learned was that by using this type of way of designing consortia, we could enable translatability from one environment to another, which I think has been a fundamental problem in the field of trying to design consortia. Basically, if you design something in one environment, it doesn't necessarily mean it's going to work in another environment. So we want to better understand how it is that we're getting invariance of function of these synthetic systems across environments that we didn't even train on. And finally, what I'll say is that we want to know whether our consortium can work in humans. We at the DFI are uniquely positioned to ask this question because we have we will have a good manufacturing practice facility in-house. So we're hoping that we can create this consortia in-house and start a clinical trial for people who are suffering from recurrent Klebsiella pneumoniae infections. So with that, I would really like to thank Rita, who was really the single postdoc who did all of the work that I showed today. I'd also like to thank my close collaborator, Sepa Kuhn, and some people from his team, Mahmoud Youssef and Kishak Lee, Robert Chen, who I knew from Jeff Gordon's lab, and Eric Paymer for providing a lot of support and being a co-mentor for Rita as she went along with this. And thanks to many of the people who pay the bills. Thank you.
All right, wonderful. Thank you. Yeah, lots of good questions on that. We'll go ahead and save them for the Q&A session. Thank you, Dr. Raman.